Hello, my name's Tim Oldham and I'm the CEO and Managing Director of AdAlta Limited. It is my great pleasure to introduce you to our company today, Securities Exchange listed drug discovery and development company. As my comments today include forward-looking remarks, I would remind everyone that these are subject to the usual disclaimers. AdAlta's purpose is to use our unique Ibody technology to create multiple novel therapeutics for debilitating diseases that have proven difficult to drug with traditional antibodies. There are 4 features of our company that I would like to share with you today. Firstly, I'll describe our Ibody platform and its ability to create therapeutics that address targets underserved by traditional antibodies. I'll describe our lead asset, AD214, where we are preparing an inhaled version for phase 2 in idiopathic pulmonary fibrosis. I'll move on to describe our 2 co-development collaborations with GE Healthcare for PET imaging in immuno-oncology and Carina for CAR T cell therapies. And finally, I'll talk about how we're building out our pipeline with additional programs to a target of 10 programs by 2023. Today, we are seeking partners for our lead asset. We're seeking co-development collaborators with a drug target suitable for addressing with our Ibody technology, and investors who are willing to join us on an exciting journey to build out a true platform company. Let's then turn to Ibodies. Ibodies are single domain antibodies. They're approximately 10% of the size of a traditional monoclonal antibody. They mimic the size, shape, and flexibility and stability of antibodies found in the shark immune system. Ibodies are a fully human scaffold that mimics that shark scaffold, and they have unique features that enable them to access challenging targets that have defeated traditional antibodies and can be formatted in a range of different ways to enable use in a diverse range of applications. We've demonstrated an immensely powerful drug discovery platform. Ibodies have been able to hit a wide range of target classes, and we're particularly interested in G protein coupled receptors, representing 30% of all drug targets for marketed drugs today, yet only 2 monoclonal antibodies approved against these targets, clearly in need of greater accessibility from an alternate antibody format. We're also demonstrating and building out a wide range of product formats. Ibodies can be used as direct therapeutics where the Ibody engages the target and induces the required pharmacological response, as is the case with our lead product, AD214. We can also use the Ibodies as a particularly defining and discerning targeting agent to deliver a therapeutic cargo more precisely. And this is what we're doing with the PET imaging agent against granzyme B with GE Healthcare. And we can also combine the Ibodies in multiple different ways to create bispecifics and multispecifics. And doing this in combination with the targeting capability of an Ibody is enabling us to generate next generation CAR T cells with Carina. So let's turn first to AD214. The problem we're trying to solve here is initially idiopathic pulmonary fibrosis, but more broadly fibrotic diseases generally. Idiopathic pulmonary fibrosis, scarring or stiffening of the lungs, is a degenerative, progressive and irreversible disease that kills most patients less than 4 years after diagnosis. While there are 2 marketed drugs today generating $3 billion in sales, they're not particularly effective, and there's a desperate need for better therapeutic outcomes, and this means new ways to new modes of action to address this disease. Unfortunately, the burden of fibrotic lung disease following COVID-19 is likely to be high, increasing the importance of developing new therapeutics against fibrotic disease. AD214 is a first-in-class treatment for fibrosis targeting the CXCR4 pathway. CXCR4 is a G protein coupled receptor, which is not only implicated in the formation of and mobilization of blood cells from the bone marrow, it's also extensively now implicated in fibrotic disease. And as you can see from the top images, the presence of fibrotic tissues, scarring and thickening of that lung tissue is associated with significant increases in CXCR4. The bottom panel shows what happens when we block CXCR4 in the gold standard animal model of lung fibrosis the mouse bleomycin model. The middle panel shows the effect of bleomycin on these mouse lungs. The blue staining here, or the purple staining here, is collagen. And on the right, when we treat with AD214, you can see that we retain much more of the normal tissue architecture with a significant reduction in collagen. This has encouraged us to move AD214 into phase 1 clinical trials. And in a phase 1 trial using the intravenous route of administration, we've demonstrated that AD214 is very well tolerated in single doses to 20 milligrams per kilogram and multiple doses to 5 milligrams per kilogram in healthy volunteers. In addition to being well tolerated, with no dose-limiting toxicities observed and no concerning immune responses, we've also demonstrated that AD214 clearly engages the CXCR4 receptor on white blood cells and in the bone marrow. And our pharmacokinetic and tissue distribution profile, while satisfactory, was coupled with preclinical results that showed we were getting significant liver localization, and this has led us to support and move towards an inhalation route of administration. I would point out that the presence of this liver localization does not detract from the efficacy results we have seen in the mouse bleomycin model by that same intravenous route of administration. We believe inhalation is a way more patient-friendly route of administration. It offers improved bioavailability, particularly given this liver localization effect. It offers significantly enhanced cost effectiveness, and it enables us, when we combine it with other routes of administration, e.g. for eye fibrosis, to partner AD214 by indication, because we will not necessarily have the crossover between indications that would occur with a pure intravenous route. So we believe that this phase 2 route we're working towards offers ultimately a better product with a better target product profile than was possible with our original IV plans. The pathway to phase 2 now in this inhalation route of administration is very well mapped out. The rate limiting step is actually the supply of the next round of GMP clinical material. We expect to be back in the clinic in the middle of 2023. And you can see a very clear set of milestones to de-risk the program as we go along the way to get there. Assets in IPF are valuable, as shown by these relatively recent transactions across the spectrum from preclinical to phase 2. And we will be looking to partner AD214 anywhere from now through to the end of phase 2. In addition to idiopathic pulmonary fibrosis, we have multiple indication extension options available to us with AD214. Eye fibrosis and skin fibrosis are obvious candidates that do not require systemic administration, and we're continuing to work on improved intravenous formulations that open up further indications in kidney disease, where we have some particularly encouraging preclinical data with an IV route of administration already, as well as liver and various forms of cancer. So now moving on to the second application of Ibodies, and this is their use in immuno-oncology to direct PET imaging agents. You'll recall or will know that immuno-oncology drugs are transforming cancer outcomes by reactivating a patient's own immune system to fight the cancer. We know that granzyme B is produced by the immune cells in order to kill the cancer, and this makes granzyme B a potential biomarker of the immune system post-activation by immuno-oncology drugs. This market is a $95 billion market, and yet only 20 to 40% of patients are responding to these drugs. So there's a burning need to be able to identify the responders early, and imaging granzyme B could well be the pathway to doing that. The market for PET imaging agents is at an attractive $6.4 billion, with the lead products in excess of $400 million. And importantly, PET imaging agents have substantially shorter development times than therapeutics, which for a company like AdAlta brings it much closer to royalty revenue streams. Our collaboration with GE Healthcare combines our Ibody platform with a leading global supplier of PET imaging agents and capability in PET imaging, conjugation chemistry, and commercialization capability. We have discovered a panel of granzyme B Ibodies binding Ibodies that have now moved into preclinical proof of concept studies at GE Healthcare. And GE have been paying AdAlta research fees and development milestones along the way. So this essentially is a pipeline asset for us at no financial cost to AdAlta. To date, we've booked approximately $1.5 million in revenue from this collaboration. The third application of Ibodies is our collaboration with Carina Biotech to develop CAR-T therapeutics and to develop next generation CAR-T therapeutics. CAR-T therapy, again, is transforming cancer by re-engineering a patient's own immune cells so they now have the capability to recognize cancer. These cells are genetically modified in the lab to insert chimeric antigen receptors or foreign receptors that recognize a tumor-specific antigen. They're expanded in the lab and given back to the patients. This takes advantage of the body's own natural immune response to pathogens. The first products were approved in 2017 for blood cancers. They already generate $1 billion in sales each year. The expectation is that more than 2 million cancer patients will be able to access CAR-T therapy within the next 10 years, which will create a $20 billion market. And more than 50% of that is projected to come from solid tumors by 2030. Now, the challenge with CAR-T therapy today is so far it's been primarily limited to blood cancers. Our collaboration with Carina Biotech combines Carina's world-leading proprietary CAR-T technologies that provide superior access and potency and resilience to tumors with AdAlta's unique Ibody platform that provides exceptional reach and targeting capability. And together we're working to create precision-engineered Ibody-enabled CAR-T therapies that provide new hope for patients with cancer. What are the synergies in this collaboration? Well, AdAlta provides, through its Ibodies, the ability to access new and difficult antigens, expanding the repertoire of potential tumor-specific antigens available to address solid tumors. This is the first limitation of solid tumor CAR-T therapies today. Further, the small size of the Ibodies confers greater precision and flexibility, enabling bispecific and dual-specific CARs to be manufactured much more easily with fewer manipulations and modifications to help enhance specificity. The second two challenges relate to the ability to penetrate the tumor mass for immune cells and the ability for those tumor cells to persist within the tumor mass once they've got there. And Carina's proprietary chemokine receptor platform and proprietary manufacturing processes are ideally suited to achieve these objectives with a best practice manufacturing process and the use of these chemokine receptors that both enhance trafficking to the tumors as well as enhancing persistence and overcoming immune suppression when they get there. So when you put all that together, you can see that AdAlta has a very solid business model that enables us to continue to build successive assets, both wholly owned, such as AD214, and co-developed, such as with GE Healthcare and Carina, from the back of our Ibody platform. In addition to the two existing collaborations we have today, we're expecting to add 2 more additional targets into our pipeline in 2021. So where does this take us as a company? We're aspiring to be a genuine platform company of the likes of Ablynx, Heptares, Receptos. These companies have progressively built platform technologies into multi-product portfolios using a combination of collaborations and internal development. Ablynx was a good example that grew from its initial clinical trial in 2007 to 10 years later being acquired by Sanofi for $5 billion US. And AdAlta today, with our first clinical trial in the bank, is embarking on a similar journey. We have a diverse and well-skilled industry experienced team driving the execution of our strategy forward. And this leaves us with what we believe is a really attractive both investment proposition and also a partnering proposition for companies seeking a new way to target difficult antigens. Our Ibody platform is ideally suited as a powerful drug discovery engine to create value by targeting difficult and challenging targets. Our lead asset is addressing a $3 billion market with phase 1 complete and a clear pathway to an enhanced and improved patient convenient formulation in phase 2. We already have 2 co-development collaborations in place with GE Healthcare and Carina Biotech. Thank you for your attention today. I'd be delighted to field questions and to have further discussions with potential partners and potential investors about our very exciting company and its journey forward.